So it's actually a bit funny that our talks have been grouped together with the previous presenter, because I'll be presenting kind of a contrasting perspective on this token-based structural alignment, and I'll be advocating, I guess, for some of the advantages of a superposition true geometric structural alignment. So before I begin, I'll give a short introduction to traditional methods for geometric protein structural alignment. And these methods are initiated by positioning structures in a shared coordinate system such that we can identify geometric similarity based on extracting pairwise distance matrices between residues of the two proteins. And then we can use these pairwise distance matrices as inputs to a classical Smith-Waterman alignment to identify corresponding residues between the two proteins, which are co-located in Cartesian space. Then once we have this initial kind of alignment, we can use this alignment to update our initial superposition by minimizing the Euclidean distance between corresponding residues. And then given this updated superposition, we can update our pairwise distance matrix and then update our alignment. And this sort of iterative process can be repeated until the method converges to a good alignment. But the major limitation of this approach is it's really sensitive to that original superposition used to start the process. So if we start with a poor quality superposition, we'll finish with a poor quality alignment, even between structures that have clear structural similarity. And the sort of the traditional approach to combat this limitation is to just try a really large number of candidate transformations with the idea that at least one of these will lead to a high quality alignment. And the way that these candidate transformations can be generated are just by extracting a comprehensive set of contiguous fragments from each of the proteins and then superimposing those fragments to generate that initial seed superposition. So my approach is quite closely related, but rather than using kind of these arbitrary contiguous fragments, I instead focus specifically on these secondary structure segments to generate a kind of a minimal set of high quality superpositions with which to start the process. And then the idea is that these initial transformations can be evaluated using a coarse-grained segment-level alignment score, which is efficient to compute because we don't have that many segments in a typical protein relative to the number of residues. And then only the top scoring transformation will be progressed to the more expensive all residue kind of refinement of the alignment. And then also I should mention at this stage, any pairs of proteins that don't meet some minimum quality threshold at this stage in the process can kind of be terminated here, because we don't really want to waste computation on proteins that are clearly dissimilar in the first place. So the other advantage of this segment-based approach is that we can use this initial segment-level alignment to constrain our downstream all-residue alignment. So in the naive approach, where we have to compute an all-by-all pairwise distance matrix, which scales quadratically with the length of the proteins, in the segment-constrained approach, each residue from protein 1 only needs to be compared with residues from the matching segment in protein 2. And since the typical segment length should be roughly independent of the size of the protein, this reduces the complexity to be kind of pseudo-linear in the length of the protein. So yeah, now we have these established heuristics for geometric protein structural alignment. It's time to benchmark. So I evaluated search sensitivity on the Scott benchmark and evaluated some representative methods from the literature. So the Scott benchmark involves pairwise all-by-all comparison of representative domains from the Scott database with structural classifications at fold, superfamily, family level. And then for each query, we can rank structures by structural similarity using each of the methods. And then performance can be evaluated based on how many of the structures are returned with the same structural classification as the query. So in this benchmark, a method called SPalign of the methods evaluated achieved the highest sensitivity, but required about 3 days to complete the 100 million pairwise comparisons. And of course, Martin's method was very fast, completed that same benchmark in under 4 minutes, but there was an appreciable drop in sensitivity on that benchmark. So there is kind of space for improvement here. So when I introduced my optimization heuristics in combination with a version of SP score, which was used for SPalign, I was able to substantially reduce the execution time of this benchmark on the same hardware to under 40 minutes, while also actually increasing the search sensitivity, at least at the fold level. And then when I push the heuristics a little bit further and trade off a little bit of the tiniest little bit of performance, I can get execution time even under 25 minutes, which is really approaching quite quick. Okay, so beyond search sensitivity, it's also worthwhile to evaluate how well these methods can, or the quality of the residue level alignments produced by these methods. And so the Homstrad benchmark contains pairs of proteins that are from the same families, along with manually curated residue level alignments. And we can evaluate methods based on how well they can reproduce these manually curated alignments. And so while developing SPfast, I noticed a clear trade-off between sensitivity and alignment accuracy, which seemed to be mediated by this empirical score used to penalize alignment gaps. So to try and work out what was going on here, I've highlighted an illustrative example. So here's a pair of proteins from the SCOP benchmark, which are drawn from totally distinct SCOP folds. So this would be considered a false positive match in the SCOP benchmark. But these structures very clearly have high similarity within a local region of the structure. And the problem is with SP score or any SP score based scheme is that it's kind of normalized by a version of the alignment length. So any high fidelity but low coverage alignment will kind of score well in this kind of paradigm. And when we don't include an alignment gap penalty, we can see we get kind of these junk matchings over here in this non-local region, which are totally unrelated to the evolutionary relationship. And this has the effect of like partially suppressing the score of this match. But it's kind of a hack. This isn't really not how we would want to handle this problem. So fortunately, SP score provides an alternative lever that we can pull to control the balance between alignment fidelity and alignment coverage, such that we can ensure that alignments that have larger coverage, but maybe at slightly lower fidelity, which would be considered SCOP true positives, are better than these local matches. And that could be controlled via this sort of alpha and D0 parameters. And so fortunately, SPfast is very quick. And so it's very easy to do a quick grid search over these parameters in order to improve performance in both sensitivity as well as alignment accuracy. And then when I transferred those parameters back to the original SP-align optimization algorithm, I also improved performance in that setting. So, but really, we don't care about these small data sets because we can be slow because they're small. What we really care about is these large databases of model protein structures. So to evaluate performance in this more realistic setting, I collected the representative domains from the AFDB clusters dataset and then searched a collection of structures against the 2 million models. Unfortunately, in this setting, there's no labels. So it's very hard to judge the quality of the search and search sensitivity. And I have used a kind of an orthogonal method to control or to evaluate, to adjudicate the quality of hits in this benchmark. And at least based on this orthogonal method, the top ranking hits returned by SPfast outperform or are adjudicated to be better quality than those returned by FoldSeek, particularly when the pre-filter is used, which is responsible for a lot of the speed of FoldSeek. So this sort of gives you some of an idea about some of this speed sensitivity trade-off. But I also wanted to highlight this additional example. This is another example of one of the queries in this benchmark. It has this beta-beta alpha repeat structural motif. And when I search against the representative structures from AFDB clusters, this is one of the top ranking hits by SPfast. And we can see by eye, it's very clearly a related structure, has the same structural motifs and the same global topology. And it is adjudicated as a good quality hit by this orthogonal method. And it is found as top, high ranking by SPfast, but it's outside the top 2000 by FoldSeek, even without the pre-filter. And so instead, what FoldSeek finds this third structure within the top 500, which is clearly a different fold. And the reason that there's this confusion is because it has this same beta-beta alpha sequence of secondary structure segments, but arranged with a distinct global topology. And so these helices that are sitting underneath this beta sheet, since they have the same local structural environment in that they are in contact with the beta sheet, they are adjudicated to be compatible with these alternative helices, even though they have a completely different global arrangement in space. And indeed, when we look at the FoldSeek alignment or the 3DI state alignment for this particular pair, we can see that it does, in fact, erroneously align these helices. So I really think this is a good method. So I wanted to make it as accessible as possible. So I've produced a PyMol plugin as well as a Colab notebook to enable high throughput search with one click. And these will be publicly available imminently after I return to Australia. And finally, just acknowledging the people that supported this work.